Hello everyone, and welcome to our AP Biology end-of-year review cram session video. If you're watching, you probably have an AP exam to study for, so let's get started. I'm going to be doing a really fast, high-level content overview of some of the hot topics you might have encountered in your AP Biology course from the end of the year. This is by no means everything you'll need to know for the exam, but hopefully it'll help you refresh your knowledge as you begin to study and cover some of the really important content that you may not have gotten to in your class yet. Today, I'm going to be covering units 6 through 8 in the AP Biology curriculum, but your teacher might have taught these topics in a different order. Whenever you're studying, there's lots of other resources on this channel that'll help you be successful in biology or other life science courses, so be sure to subscribe if you don't want to miss out, especially on this year's last-minute crash review AP Biology video. Now, as a reminder, DNA, one of our most important nucleic acids, is found in the nucleus of eukaryotic organisms or in the nucleoid of prokaryotic organisms. DNA stands for deoxyribonucleic acid. We're also going to talk about ribonucleic acid or RNA. So again, one nucleotide contains a phosphate group, our deoxyribose sugar, as well as our base, which can be A, T, G, or C in DNA. Here again, we're looking at many nucleotides put together. Our phosphate here, deoxyribose sugar, and our base. Now, importantly, when we look at this, our adenine and guanine are a little bit different from cytosine and thymine. Adenine and guanine are purines, meaning they have a double ring structure when we look up close. Pyridamines, which contain thymine and cytosine as well as uracil and RNA, have a single ring structure. Now, adenine and thymine are pairing together with 2 hydrogen bonds. Cytosine and guanine pair together with 3 hydrogen bonds, but these are easily broken during DNA replication or transcription. This is something you'll absolutely have to memorize for any biology class. So A always pairs with T, G always pairs with C when we're talking about DNA molecules. Some people like to remember this as apple tree good cookie or all teachers can go if you do it backwards. Whatever mnemonic that you have to remember this, make sure that A's and T's and G's and C's always go together. Another one that I've heard before that is helpful is that capital A's and T's are written with all straight lines and capital G's and C's have those curvy shapes, which is another way to remember which ones go together. Here are purines and pyridamines up close. You can see that adenine and guanine have 2 rings. Pyridamines have 1. A great way to remember this is pure as gold, cut the pie. This is a mnemonic to help us remember that our purines, pure as gold, are adenine and guanine, as seen as the as gold, A and G here. And our pyridamines are cut the pie, C and T are in the word cut, pie for pyridamines, PY. Hopefully that'll be helpful. Now let's talk a little bit about RNA. Remember, RNA is single-stranded. It's not going to have that double helix that DNA does. Both RNA and DNA do have our 3 components in our nucleotide, a sugar, a phosphate group, and a nitrogenous base. And these are going to form units connected by covalent bonds with 5' and 3' ends. But when we look at the sugar in DNA, that's going to be a deoxyribose sugar, hence the name, and RNA is going to have a ribose sugar. These are both pentose sugars with 5 carbons, but they're a little bit different. DNA is going to contain thymine, whereas RNA contains uracil. So instead of having a thymine, RNA is going to have that U. DNA will always have T's. DNA is usually double-stranded unless we're dealing with certain situations, and RNA is usually going to be single-stranded. DNA doesn't really leave the nucleus in eukaryotic organisms. RNA will leave the nucleus as mRNA or rRNA or different types of RNA for protein synthesis and other functions within the cell. Now, one nucleic acid we don't want to forget is ATP. Adenosine triphosphate, you might remember from studying cellular respiration in biology. ATP is also a nucleic acid, and you notice it has a very similar structure to DNA. We have phosphates, we have sugar, and we have our base. But unlike DNA or RNA, ATP has 3 phosphate groups attached to the sugar, and this is also a ribose sugar. And energy is formed from ATP when these phosphate groups are broken, and we get adenosine diphosphate instead of triphosphate. Chromatin, or the DNA that is in the nucleus in a eukaryotic organism, is typically wrapped around proteins called histones, which then group into something called a nucleosome. So we have lots of DNA base pairs wrapped around a core of histone proteins, and then these nucleosomes themselves are organized into different structures so that we can finally package it into chromosomes if necessary. Talk a little bit about DNA replication. Remember, it occurs during the S phase of our cell cycle to allow daughter cells to have an exact copy of the DNA after they go through cell division. If they didn't duplicate before they went through cell division, we would have half the DNA each time the cell divided, and soon almost no DNA at all. Now, with the help of some different enzymes, the DNA molecule is unzipped or opened up, and then we have 2 strands that we can add new nucleotides to. We'll have one old side of the DNA and then one new side in each of our 2 new DNA molecules. So remember, it's split open by our enzyme helicase, and then we'll add new nucleotides to each of those open sides, ending with 2 new molecules or 2 new DNA strands, each half old, each half new. This is why we call it half conservative, semi-conservative. Some of our important enzymes that we're going to talk about are helicase, polymerase, ligase, and topoisomerase, which is a mouthful. It's an enzyme that helps stabilize the DNA during the unwinding, so we don't have any supercoiling or tight twisting and coiling of our DNA. So it's a little bit ahead of where we're actually being unwound. Helicase I don't have depicted here, but this is going to be our unzipping or our molecular scissors that'll actually unzip our DNA. Polymerase is going to add to our template strand by building the new strand, so adding the new nucleotide sequences on or as a complement to the template strand. And then ligase is going to covalently connect sections of DNA that are being synthesized in fragments. So our DNA ligase is important here, especially when we get to our lagging strand or our Okazaki fragments. Now remember that replication is a semi-conservative process, so one strand serves as a template for the new complementary strand. Each strand is anti-parallel, which means the 5 to 3' direction of one strand runs counter to the other 5 to 3' direction of the other strand. So if we have a 5' on this end, we would have a 3' on this end. And remember, those primes actually refer to the carbons in the sugar in that nucleotide. We say DNA gives us our traits. So how do we get from a strand of DNA all the way to an actual trait, like the color of our eyes? Well, it all depends on proteins. And so protein synthesis is the process of how the message of DNA is transcribed and then translated into proteins. So synthesis just means making. So if you're trying to remember what protein synthesis is, just think about how we get proteins, how we make proteins from DNA. Let's see how this works. So remember, the DNA is in the nucleus of eukaryotic organisms. So we have our DNA strand, it's going to be split apart, and it's going to start to be copied as mRNA. And so this mRNA strand will build, basing itself off of part of that DNA strand. There's a couple of differences between mRNA and DNA, and one of those things is that instead of T's, RNA has U's. So when we pair with A, we're going to match it up with a U instead of a T in mRNA. RNA is also single-stranded, so once it's done copying the DNA, it'll leave the nucleus, the DNA will go back together, and then the mRNA will reach the ribosome where we can do the rest of the work, the translation part, where we're going to get our tRNAs, which are another type of RNA, are going to match up with the message on the mRNA and bring over these little molecules called amino acids. So glue here, that's one amino acid. And as these tRNAs match up with the code on the mRNA, we're going to bring more and more amino acids over, and they will be linked up by bonds here. These are peptide bonds, and we will start to form a protein. And once we have our amino acids linked together, we have our polypeptide or our many peptide chain, and that will result in our protein, which will do the job for the cell that we need it to do. It gets very long and it folds up in a specific shape, so it becomes a functional protein, like an enzyme, for example, or a pigment protein that'll color our eyes. So again, some differences between DNA and RNA. DNA is double-stranded. Think of that D for double. RNA is single-stranded. DNA has actually a different sugar than RNA. It's a deoxyribose sugar. RNA has a ribose sugar. And then, of course, DNA has T's, where RNA has a U as one of its bases. So let's practice here. If you want to pause the video and write down the mRNA sequence you think would go with this DNA strand, go ahead. I'll give you a second. And here it is. This is the RNA sequence that would come from this DNA strand. So now we have a message in our mRNA that can be taken out into translation. Now, if you confuse transcription and translation, remember transcription, we're changing from one type of nucleic acid, a deoxyribonucleic acid, into another type of nucleic acid, a ribonucleic acid. We're transcribing it into a message. So thinking about you writing down a message that you're going to pass to your friend, that's transcription. Translation, on the other hand, if you're translating something from English to French, you're changing languages, and that's what we're doing here in the cell as well. We're changing molecular language. And so we're going from a nucleic acid to an amino acid chain or a protein. So if you want to remember which one's transcription and translation, think about which one's writing down the message, that's DNA to mRNA, and which one is getting us our chain of amino acids, or we're changing our language. We're going from a nucleic acid to a protein, so that's translation. Now, you might be asked to read something like this called a codon chart or an amino acid table, where we're given all these letters here and it tells us which amino acids go with which 3 groups or codons of RNA. So for example, if our codon was UCA, we would start here in the center with the big letter U, and then we'd find the C in the next level, and then we would go to the outermost layer, A. Okay, so that's serine, or SER. This is an abbreviation for an amino acid. Let's try another one. How about AAA? So we start here in the center, A, and then we look here to see which one this touches. A, that's lysine. That's another amino acid. So every codon matches with a specific amino acid. Now there's other types of codon charts as well. You may see one that looks like this. You have to read them a little bit differently, but sometimes they're broken down very helpfully. All right, let's review that one more time because there are a lot of steps, and this time I'm going to add in a few more extra details that'll help if you are an AP or upper level biology student. Hopefully you remember a little bit about protein synthesis, where the enzyme RNA polymerase is going to synthesize mRNA molecules in the 5' to 3' direction by reading the template DNA strand in the 3' to 5' direction. So we have our RNA polymerase synthesizing that molecule, the mRNA leaving the nucleus, and then undergoing certain modifications, which we'll get into a little bit later. And then in the cytosol at the ribosome, we have our tRNA bringing around our amino acids, and it recognizes each codon with an anticodon on the tRNA, and that amino acid chain is built from the mRNA transcript. In prokaryotic organisms, this happens all at once. Transcription is coupled to translation, but in eukaryotic organisms, we have this occurring in our different locations. Once we have our amino acid sequence, we can undergo even more protein modifications to get the final functional protein. Talk about mutations. Mutations can be caused by mistakes in replication, transcription, translation, or other parts of the cell cycle. They can also be caused by mutagens, things that are going to change up the DNA order, things like X-rays, UV radiation, chemicals, etc. And some of the effects of mutations, they can have no effect at all, and we call those silent mutations. The changes could be good. They could introduce a new trait within an organism, or they could be really bad, or it could even cause a genetic disorder or death within the organism. So it really depends on how severe the effect is. A mutation that introduces a new base, for example, adds in a little C in the middle of a DNA chain, might have a huge effect on the protein, because then everything in the reading frame gets shifted down, so that makes it a little bit more difficult to create the same protein that we wanted originally. So if you think about some questions that you might get related to mutations, you might be asked, like, what would likely cause an increase in the frequency of genetic mutations, and something like exposure to X-rays would be a good answer for that. So understand that mutations are changes in DNA coding, and they can be deletions, additions, substitutions, and they can be random or spontaneous, and they can be caused by lots of different things. Okay, so when we talk about gene expression, we're talking about how we're going all the way from DNA to protein, and how much protein, what kind of protein is produced from different sections of DNA. And this can look really different depending on the level that we're looking at in this full process of protein synthesis, or in prokaryotic versus eukaryotic organisms. So let's take a look at prokaryotic organisms first. There's 2 main types of operons that we're going to talk about today. Inducible, like the lac operon, and repressible, like the trip operon. Inducible operons are basically ones that you can turn on, and repressible operons are groups of genes that you can turn off. And when we say turned on and off, what we mean is transcription, or the action of RNA polymerase transcribing genes or sections of DNA into mRNA that will later be translated into proteins. Let's start with inducible operons, or something like the lac operon. In its usual state, we do not have transcription happening. That's mostly because we have a repressor here that's bound to a section called the operator in this operon. Now, the purpose of these genes, LAC-Z, LAC-Y, and LAC-A, is to provide instructions for the cell to build parts of proteins to digest lactose. If there's no lactose in the cell, these genes don't need to be turned on because we don't need any enzyme to digest lactose. But when lactose does show up, a version of it, allolactose, can bind to the repressor, removing it from the operator. This allows RNA polymerase to move down the operon, and transcription can occur. Once transcription occurs, we have the lac mRNA, which can then later be transcribed into a protein that can digest the lactose. Once that lactose is all gone, it no longer binds to the repressor, which means it's back on the operator and we cannot have transcription any longer. So let's apply this. Let's say there's a mutation in the gene that codes for our repressor protein. Remember, the repressor is a protein itself as well. And in that mutation, allolactose can no longer bind to the repressor. What happens to our transcription? Well, if allolactose can't bind to the repressor anymore, that means the repressor stays on the operator because the repressor will not be removed unless allolactose binds to it. When allolactose can no longer bind to the repressor and the repressor stays on the operator, RNA polymerase can no longer move down the operon and transcribe the genes to generate these proteins. So we have low or no levels of transcription. Let's take a brief look at a repressible operon. This is a type of operon that's going to keep transcribing or keep producing a product until it's turned off. So in its usual state, it is transcribing and creating these proteins which will generate tryptophan, something the cell needs. But if tryptophan is present in the cell, we don't need to build proteins to make that tryptophan itself. So tryptophan will bind to a repressor, bind to the operator, and then prevent RNA polymerase from transcribing the rest of these genes. Remember, operons are a type of gene regulation in prokaryotic organisms, and these groups or genes are transcribed as a single mRNA molecule. And we can have both inducible operons, ones we can turn on by removing repressors, and repressible operons, ones that we can turn off by adding repressors. Now, in eukaryotic organisms, we do not have operons. Instead, we have many levels of regulation for gene expression. There's pre-transcriptional regulation, which involves several things before transcription even occurs, especially how accessible that DNA is itself. Remember all of that DNA organization we talked about how it's wound around proteins in its chromosomes? Well, if it's folded up in certain ways, it's hard for our enzymes and other transcription factors to access it, to actually transcribe it. So we have special complexes of molecules that can regulate the availability of parts of this chromatin in order for transcription to actually happen. Also, we're going to have different transcription factors that can help transcription get started in different ways. All of this is going to happen before transcription even begins. At the transcriptional level, we can have things like promoters or enhancers that are going to help regulate the initiation or the beginning of our synthesis of RNA. And then after transcription, so post-transcriptional regulation can involve lots of different things. One of the most important that we'll talk about in a little bit more detail is alternative splicing or removal of introns. So these are different sections of DNA that are non-coding that are going to be taken out of the mRNA before it goes on to translation. We can also have alternative splicing, which we'll talk about in just a moment. Additionally, that mRNA can have parts modified or added, like the addition 5' cap and something called a poly-A tail. That's going to help with the stability of the mRNA molecule as it leaves the nucleus. And then once it's out of the nucleus in the cytoplasm, we have things called microRNAs, and they can bind to the mRNA molecule and can either prevent it from undergoing translation or even degrade it. And if that mRNA is degraded, we won't have the expression of the protein that it's coded for. Then that mRNA can be sent to different parts of the cell where there's different ribosomes that are going to translate the mRNA at different levels. So that's called mRNA localization, and all of these things are going to play a role in how much protein is produced in the cell. After translation happens, so the mRNA has arrived at the ribosomes, we have our amino acids joined together through peptide bonds. There can be modifications of those proteins like phosphorylation or even degradation too. Those proteins can be broken down by proteasomes. And so all of these factors together can contribute to the regulation of gene expression in eukaryotic cells. Let's go back and talk a little bit more about that alternative splicing, which is one of our post-transcriptional modifications. So when a gene is transcribed to make mRNA, we're going to have a copy that includes what we call coding segments and non-coding segments. Coding segments contain instructions for making those proteins, and the non-coding sections are what we call introns, and they are unnecessary for the actual assembly of the protein. So we don't need those parts, and so those parts are going to be cut out or removed. So then we get a modified mRNA that is a little bit shorter and contains all of the coding sections. Now we can also have what's called alternative splicing, where we only include some of those exons, or we can have exons appear in alternate formats from the same initial gene or the same initial mRNA. So for example, these two mRNA sections here could both come from the same initial template, but have different sections be cut out, and the result would be a different type of protein. One way to remember this and keep this straight is to think of the mnemonic: introns go in the trash and exons are expressed. So in the trash for introns, we cut those out, exons are expressed. But remember, we can express these alternatively, even though they're going to stay in the same order. So we're never going to flip sections 1 and 2, for example, here, but we could go 1, 3, 4, 5, or we could go 1, 2, 4, 6, depending on what's necessary for the protein. All right, let's take it back to prokaryotic gene expression and compare the two really quickly. In prokaryotic gene expression, a lot of that is going to rely on operons. We can have one promoter for multiple genes, and there are no introns. Some sections can coil, but there's no nucleus, and so we have transcription and translation sometimes taking place in the same locations within the cell. In eukaryotic gene expression, we can have these genes that are spread out along long linear chromosomes. We have one promoter for one gene. There are many introns that are removed, and we can have exons that are spliced to create that mature mRNA that's going to go and then be translated into a protein. It's highly organized. We have our chromatin wrapped around histones, and there are additional control mechanisms because transcription and translation are taking place in different locations. Remember, transcription is in the nucleus, translation is in the ribosomes in the cytoplasm. So biotechnology can mean a lot of different things, but mostly when we say biotechnology in biology, we're referring to the modification of living things to create products for humans that are useful to us. Very commonly, these are things that are used in medicine or healthcare. You might have heard of the CRISPR-Cas9 technologies and how all of a sudden we have sickle cell treatments on the market now because of this biotechnology. But with any kind of rapidly evolving technology, there are going to be questions that come up. And so one of the things you might hear talked about in your classes is the ethics of biotechnology, or the ideas behind what we should do or we shouldn't do, and ethics aren't always black and white, so it's really good to talk about these things and discuss pros and cons and societal implications to any new technology that arises. So for example, we have mosquitoes that we can release into populations that have been modified to be sterile or to produce sterile offspring when they mate with living mosquitoes in a population. This can reduce the spread of disease, but maybe this could have unforeseen effects on natural populations. There's also so many easily available at-home genetic testing kits that have a huge range of implications for society and the general public. There are so many different techniques within the realm of biotechnology. If you ever take a course in biotechnology or go on to study more biology, you'll find this out. But there's a few that are focused on in AP biology. These are PCR, gel electrophoresis, DNA sequencing, and bacterial transformation. All of the smaller details of how to do each of these techniques is beyond what the exam is actually going to test you on. As long as you have a good conceptual understanding of each of these techniques, you should be in a good place for the AP Bio exam. Now we're going to focus mostly on PCR, gel electrophoresis, and genetic transformation today because DNA sequencing is not covered very frequently on the exam, and it's changed so much from the days of Sanger sequencing all the way up to next-gen sequencing, which a lot of scientists use now, that you really don't need to get into the details of this for AP Bio. Basically, what you need to know about DNA sequencing is that it's the process of determining which nucleotides come in which order in a sample of DNA. Now we can do this at a much lower cost, and we're able to sequence short pieces of DNA really, really quickly for study in modern labs. Sequencing an entire organism's genome is still a little bit tricky, but in the future we may have the ability to sequence every individual's complete genome in order to personalize and tailor medicine towards them. But that is a topic for the future. Let's get back to PCR. So the main idea of PCR is making lots of copies of DNA from a small sample. With PCR, we can make a lot of copies in a relatively short amount of time. What you need to know about PCR is that there's special machines that you can put your DNA sample after preparing it into, and then those machines go through cycles to do these three things. First, it heats up the DNA, which denatures it or separates it into two different strands. Then we're going to cool a little bit to 55 degrees Celsius, and primers, which have been added to our DNA sample, which are these short fragments of DNA, will be able to anneal or connect to the separated DNA fragments. From there, we're going to heat it up a little bit more so that we have more of an affinity for elongation. More nucleotides, which are floating in our mix, can attach to the open fragments of our DNA. So after one of these cycles, we're going to get two new strands of DNA. Then it goes through the same process again. They're denatured or separated. We have our annealing process, and then we have our elongation. And you see how each time we're able to double the amount of DNA. So PCR just stands for polymerase chain reaction, and we'll do this over and over and over again until we get enough copies of our DNA. After that, we can use this DNA to do a multitude of different things. One very popular use is getting a DNA fingerprint or doing gel electrophoresis. Now, hopefully in your 9th grade biology class, you learned that this is not a fingerprint actually from your finger, but instead it's a particular pattern of your DNA after we run it through a gel. The first step in gel electrophoresis, or getting a DNA fingerprint, is going to be cutting that DNA with restriction enzymes. Restriction enzymes are special enzymes that cut DNA at specific recognition sites that are usually only a few base pairs in length. Sometimes these are palindromic, meaning they read the same forwards and backwards. But after that cut is made, we'll get what's called sticky or blunt ends at the edges of our DNA. And a sticky end is when we have bases that are unpaired after a cut. So let's see what that means. So we'll have our sequence of DNA. The restriction enzyme will recognize a specific sequence and cut along that site to give us two separate fragments of DNA. For example, BAMH1 cuts our DNA between the 2 Gs in this sequence whenever it encounters it along the strand. After we've prepared our DNA by cutting it with restriction enzymes, then we can put it in a gel. Our gel is a matrix-like material with tiny little holes in it that's going to allow our DNA to travel through it. Usually we put a marker or a sample of DNA that we know the exact fragment lengths of in the very first well, which is at the top of our gel. And then the other DNA samples that we're analyzing are going to be loaded into the next wells, usually with a dye, and we're doing this with a micropipette. Once we've loaded all of our samples in, what we'll do is we'll attach electrodes. So we're running charges. Up here, this is a negative charge, and down here, this is a positive charge. One very important thing to remember about the properties of DNA is that DNA is negatively charged. So if we run an electric current through this gel, DNA will start to travel towards the positive end. But remember, in each of these samples, we've chopped our DNA up. So as it travels down towards the positive end of our gel, we're going to see the DNA start to separate because each of these fragments is actually a different length. Our marker here is used for estimating the size of our DNA, but one thing we know for sure is that as the DNA travels down the gel, the longer fragments, or the larger ones, are going to stay closer to the top or closer to the wells, and the smaller fragments are going to be able to travel farther down the gel. So when the electric current is applied, the negatively charged DNA is going to move towards the positive end of the gel. But in each sample, each of these fragments are going to be different lengths, so we'll see different bands for the separation of the DNA molecule. One way I like to remember this is that if you have a race between one really, really large, probably out of shape person and one really, really tiny, pretty nimble, fit person, the person who is smaller is going to be able to run faster and win the race. And so that is what this DNA is doing. They're able to get through all of these holes in the gel faster because it's a smaller fragment and a smaller piece. You should be able to know the closer this is to where the wells are, the larger it is, the closer our DNA is to the positive end of our gel, the smaller it is. So now that we know we can separate DNA based on size, what can this help us with? Gel electrophoresis has a lot of purposes, from DNA analysis at a crime scene, paternity testing, looking at how closely two organisms are related, evolutionarily. But let's back it up to a crime scene. If we have, for example, our evidence from the crime scene, which is DNA, and we want to compare it to our suspects that we have in our case, what we can do is look at the banding pattern, or the bands from the evidence DNA, and compare that to the bands of the suspects. Based on these banding patterns, we can see that suspect 3 is a really good match with our evidence, and we might have just solved the crime. We'll also use restriction enzymes and gel electrophoresis when we're doing genetic transformation. And what we're doing with genetic transformation is inserting a gene of one organism into another organism so that it can display a new trait. Because of the central dogma, we're able to do this because virtually all organisms use the same universal genetic code. A lot of times we'll do this with bacteria and plasmids. But what is a plasmid? A plasmid is a circular double-stranded piece of DNA that's usually found in bacteria. It's extrachromosomal, meaning it doesn't belong inside the bacteria's chromosome, but bacteria will express or produce proteins from the DNA in these plasmids, and they'll replicate the plasmids whenever that bacteria divides as well. We use genetic engineering in a lot of fields, including agriculture, for example, inserting genes for pest resistance or fungal resistance into certain plants, in bioremediation, where we've genetically engineered bacteria to uptake oil at oil spills, or even in gene therapy in medicine. We've even used bacteria to produce insulin for humans before. So how do we get an organism like bacteria to actually express a new trait that wasn't in its DNA before? Here we're looking at a sample of DNA that's been added to bacteria to make it glow green. So a very simple version of this is that we start with a plasmid, and we're going to cut it with that restriction enzyme. Our foreign DNA, or our DNA from the other organism, is going to be cut as well, and then those sticky ends will attach to give us recombinant DNA here, meaning DNA from one organism has taken up DNA from another, and DNA ligase is going to join our sticky ends together to form our recombinant plasmid. Once we have a recombinant plasmid, we'll go through several laboratory techniques to make this bacterial cell competent, meaning it'll take up that new plasmid, and once it does, we say that it's transformed. Now after that, we're going to give this bacteria a little time to grow and reproduce, so that we can see actually on plates whether or not it actually took up that gene successfully and was able to pass it on as it replicated and divided. A lot of times we'll do this by adding a resistance gene. So for example, ampicillin is an antibiotic that kills off bacteria. So if the bacteria doesn't have a gene of resistance in it already, it will not grow or it will die on the plate. So you might see a chart like this. Charts like these are very common when we talk about experiments with transformation. But the idea is that we'll set up several different plates, and with one set of bacteria, our control, we won't add the ampicillin resistant gene. In another set of bacteria, we will transform and we will add an ampicillin resistant gene to it. Then we'll wait for the bacteria to grow and see what happens. On some of these plates, we've added ampicillin, the antibiotic that kills bacteria. On some of these plates, we haven't added any ampicillin. So this normal bacteria that we haven't changed should grow on the plate with no ampicillin because nothing's killing it, and it shouldn't grow on the plate that does have ampicillin. And so this shaded in color here just means there's a bacterial lawn, which just means bacterial growth that covers the entire plate. Now let's look at the genes that we did the transformation on. For our bacteria that we actually added the new ampicillin resistant gene to, on the plate with no ampicillin, we should still see a lot of growth. On the plate with ampicillin, we're going to see growth too. It may not be as much as the plates without ampicillin because not all of the bacteria successfully transformed, but the ones that did are going to be the ones that grow and show up on our plates. So here, even though we have the bacteria killing drug, if the bacteria successfully took up this ampicillin resistance gene, it should be able to grow here on this plate, and that's what we see. So there's a lot of different versions of charts like these whenever we're doing transformation experiments with bacteria, but just be able to recognize when we're adding in genes, you should be able to hypothesize what should show up where, and justify why we have growth on certain plates like these. So when we talk about natural selection, we're talking about it as a mechanism of evolution. Natural selection is one of the major mechanisms of how organisms evolve on Earth. And evolution we can define as the change in genetic makeup of a population over time. Small changes can accumulate into massive ones over many millions of years and millions of generations. And natural selection explains how that can happen in different environments. When we talk about how an organism is evolutionarily fit, that is not about being strong or being big and bulky or fast. It's about how well they survive, reproduce, and pass on their traits to their offspring. One big deal to talk about in natural selection is the environment and how the environment can change and act as a selective mechanism on populations. And we'll get into some examples of that soon. Another key term that we're going to be talking about is adaptation. We have one definition here on screen. Another way to describe adaptation is a trait that increases fitness. So a trait that's going to help an organism survive and reproduce and pass their traits on to the next generation. Charles Darwin, as you might have heard before, is our father of modern evolutionary theory. His theory of natural selection was supported by evidence from many different scientific disciplines. He was an English naturalist, and he went to the Galapagos Islands, made a lot of observations, did a lot of critical studies, and his theories and his ideas behind natural selection really became the foundation of modern evolutionary studies. So these are the key components behind his ideas in his theory of natural selection. First, species can vary. So within a population, we can have individuals with different versions of traits. We know this when we were studying genetics. We saw there are different phenotypes within one particular population for a particular combination of genes. There is also the idea that species can change over time, and this is in relation to the populations. And last, there is a struggle for existence. So you might have heard the term before, survival of the fittest. And this just means that there's a competition for resources like space or food or light within a particular environment, and the individuals with the more favorable variations of a trait, so the best phenotypes for that environment, are more likely to survive and then produce more offspring and passing their favorable traits on to the future generation. And what else is important? We need a diverse gene pool for a population in order to survive and not go extinct. Environmental conditions can change. Something that is favorable in one environment could not be favorable in the future because of the environment changing. And we'll go over an example of this a little bit later in the video. This is a very important distinction I want you to make too. Individuals do not evolve in biological terms. One individual does not change its genetic traits within its lifetime. Populations evolve. So a group of organisms can change the frequency that a certain trait appears based on changes in reproduction and survival. But organisms themselves do not evolve. One singular organism cannot evolve. We measure evolution as the change of a frequency of a trait within a population. So make sure you write this down. Populations evolve, individuals do not evolve. All right, so again, genetic variation can come through mutation, genetic drift, sexual selection, artificial selection, and I wanted to really quickly introduce you to the peppered moth scenario. So this is a classic example of natural selection and variation and how a change in environment can actually affect the success of particular organisms. So the peppered moth is one species, and within this story, there are types of peppered moths that have sort of this lighter coloring, a little speckled, peppered, as you see, and then there are variations that are darker or black. Now, at first, the ones that were more successful were the ones with this peppered coloration because they could blend in easily and camouflage on the bark of the trees where they liked to sit. And the ones that were this sort of darker color or solid color were easily spotted by predators, and there were not as many of them being successful within the population because they were easily captured and attacked and eaten. Now, when the Industrial Revolution occurred, a lot of the trees became dark with soot from different industrial plants, and guess what happened? The organisms or the versions of the peppered moth that were darker were more likely to survive and reproduce because the lighter ones could then be spotted by predators. So what we saw was a change in the frequency in the population of the organisms that were able to survive and reproduce.
The concept of biological evolution uses lots of different types of evidence in order to support our understanding of it.
Now we can use other biochemical evidence to help us determine which proteins in DNA are similar in different organisms. This helps us construct our tree of life and gathers more evidence for evolution, helps us figure out how different organisms are related to each other. We can also use embryonic development, fossil evidence, or morphology or different physical characteristics to determine which organisms might be related and where they came from. We know that Earth's present-day species developed from earlier distinct species. When we get new combinations of existing genes or mutations in genes, and these are inherited, they can be passed on to offspring, and we can see variations in populations. For example, if there's a random mutation in this DNA where this A is changed into a G, this could then produce a different amino acid, which could then lead to a different function of a protein and give us a different trait. Mutations can be caused by different mutagens like radiation or chemicals, but they can only be passed on to offspring if they occur in sex cells. If we have mutations occurring in body cells, they're only going to be passed to other body cells that arise from the mitosis or the cell growth of current cells in the body. Over time, organisms that are better adapted to their environment are more likely to survive and reproduce and pass on their genes to the next generation. For example, if we have a population of bacteria and some of those bacteria are naturally resistant to antibiotics, when we treat that population with antibiotics, only the ones with the resistance will survive. Then that gives way to give the opportunity to the resistant bacteria to survive and reproduce and pass on the resistant gene to their offspring. And what we've done here is created an antibiotic-resistant population. The resistant bacteria were more fit, and they have survived and reproduced at a greater rate than non-resistant bacteria. When there is more variation within a species, and a species is more genetically diverse, they are more likely to survive when there's changes in the environment. When we have different types of adaptations, these can lead to the survival of different organisms. We know that behavioral adaptations, structural adaptations, and reproductive adaptations have helped different organisms survive through hundreds and thousands and millions of years. Remember that there's no one ultimately evolved organism. Environments are constantly changing, and so populations will continue to change. Also remember that individuals do not evolve, populations evolve, and evolution is just the change in different frequencies of different alleles over time. Sometimes we can have extinction of a species when we don't have adaptive characteristics that are sufficient for survival. We do see a lot of extinction in the history of life on Earth, and most of the species that have ever existed on Earth are actually now extinct, and we can tell this by the fossil record.
What is artificial selection, otherwise known as selective breeding? As far as genetically modifying plants, a lot more people are comfortable with the term selective breeding as opposed to genetically modifying something. But humans have been selectively breeding or artificially selecting organisms for thousands of years, ever since agriculture started, really. This has given us the ability to produce crops with higher yield, more resistance to things like pests or severe weather, and have better taste. So in this video, we're going to go through a few examples of artificial selection or selective breeding and talk about what you need to know about this topic. First of all, we have to get straight, as an important theme within anything we talk about with evolution is that there is variation within populations. In humans, not everybody has the same hair color or or eye color or height. Same thing goes with plants or animals or anything else we're thinking about selectively breeding. Think about apples, for instance. There is a variety of different traits within apples for taste, shape, color, etc. And through that variation, we're able to select the traits that we want and have those organisms breed and pass on their traits in the next generation. Now, we can get genetic variation in a population through a variety of ways. We've got mutation, genetic drift, and sexual selection, as well as artificial selection. But in this video, we're talking about artificial selection. And remember that evolution is the change in the genetic makeup in a population over time, not just the changing of one organism. Individuals don't evolve, populations evolve. So, humans can impact the diversity within a population, either making it more diverse, less diverse, or favoring a certain trait for our benefit. The way we do this is by controlling which organisms reproduce. In plants, if we're growing in a greenhouse or in a farm, that's easy because we can select the individuals we want to continue on to the next generation. In animals, it's determined by which organisms we're allowing to breed, but artificial selection involves when reproductive success is determined by human or our requirements. And we see this in foods, we see this in dog breeds, so we'll talk about that next. You probably have realized that dogs are very different from one another when it comes to different breeds. All dogs are the same species though, and scientists have actually analyzed the differences in the genetic makeup of all these different dog breeds and found there's actually only a few genes that control all these different phenotypes we see when it comes to the differences in dogs. So even though dogs looked extremely different on the outside, genetically they're not that different from one another. That's why different dogs of different breeds can still breed with each other and produce viable, fertile offspring. But we have artificially selected dogs for particular traits in order to produce all of the different breeds that we know today. Some people originally selected dogs for their speed or friendliness or size, but then we wanted to select for other physical characteristics. And that's how we end up with interesting breeds like you see here. Now let's talk about bananas. Bananas look very much alike and taste very similar from banana to banana. The most commonly sold banana in stores today is actually called the Cavendish banana, and all Cavendish bananas are genetically identical to one another. They're actually clones of an original banana that was selectively bred back in the 1800s. Originally, bananas that were grown and found on Earth were very hard to peel, had very tough seeds to get through, and were not as sweet as the bananas we know today. After a while of selective breeding, we chose bananas that had smaller seeds, that were sweeter, and that were much easier to peel, and eventually arrived at a variety of banana that we know and eat. That type of banana was then cloned via early plant cloning techniques, not like what they did with Dolly the sheep, and now we have genetically identical bananas across the globe. Unfortunately, this can give rise to susceptibility to certain diseases. This means that one genetic disease or one particular disease could kill off a whole plantation or a whole population of bananas really quickly and easily because there's a lack of genetic diversity within the population. Remember, increased genetic diversity within a population is actually going to increase survivability for that population and make that population more likely to survive any environmental changes, diseases, or other disruptions. Certain traits in cows we have also artificially selected for. For example, milk yield, milk production over the years has increased in certain dairy cows because we select the ones that produce the most milk and choose those to go on and breed to produce more for the production of more milk. Now, you might have seen a lab investigation in your class involving plants within the Brassica genus. These are plants that are fast growing and easy and have some easy to observe phenotypes like height, for example, particular types of flowers, or trichomes along their stems, and these are tiny little hairs that you can count from generation to generation. You could potentially select the density of the trichomes in a particular area. You select the plants that have the most trichomes and choose those as the ones to breed. After you breed those plants and watch the next generation grow up, you might be able to see an increase in the average of overall trichomes in that population. That's artificial selection right there. You could also potentially do this with height or any other quantifiable trait. Now in this video, I'm also going to mention convergent evolution, which is not always a type of artificial selection. In fact, it's its own type of evolution. This is when we see similar selective pressures that lead to similar phenotypes in very different populations or different organisms. One of the most commonly used examples of this is how birds and bats both evolved flight separately and independently. What happens is that environmental pressures are going to cause certain traits to be more favorable within a population, and then those individuals will survive and reproduce. So we could see this in body plans of certain species. We could see this in drug resistance in certain pathogens. Again, that's human action because we've created those drugs. We've seen this in antifreeze proteins in particular fish, both in the Arctic and in the Antarctic, two different species, two different locations, but similar selective environmental pressures are going to produce a similar type of protein for these fish to survive. Now, there's a lot of human influences that could cause convergent evolution to occur as well, even though it doesn't have to be caused by something from humans. Some examples are pollution, urbanization, global warming, again, domestication and agriculture, an introduction of medicines, and emerging diseases that are caused by humans. So even though this isn't, again, type of artificial selection, convergent evolution could occur because of human environmental impacts. We'll talk more about this when we start talking about human impacts on the environment, but be thinking about all the ways that humans drive evolution, including artificial selection and causes that would lead to convergent evolution. I hope this video has been helpful. Thanks so much for watching, and if you have any questions about artificial selection or evolution, please put them in the comments below. I'll see you later.
Now, what is the Hardy-Weinberg equilibrium anyway? Well, Hardy-Weinberg is actually an equation that we can use to determine the frequency of alleles in a population at a given time. Hardy-Weinberg is used as a model for describing and predicting allelic frequencies in non-evolving populations. Remember, it represents ideal populations that aren't changing. Since natural populations do change, Hardy-Weinberg provides us with a baseline to gauge that degree of change. Now, there's two equations you're going to need to know in order to do Hardy-Weinberg problems. The first one is easy. It's just P plus Q equals 1. The second one is P squared plus 2PQ plus Q squared equals 1. Now, what do each of these things represent? So P is going to equal the frequency of the dominant allele in a population. Sometimes we represent that with a capital letter, like big A. Next up, Q is going to represent the frequency of the recessive allele in a population. So for example, little a. P squared is the frequency of homozygous dominant genotypes in the population. So for example, big A. 2PQ is going to be the frequency of the heterozygous genotype in the population, so big A, little a. And then you guessed it, Q squared is going to represent the frequency of the homozygous recessive genotype in a population. So if you're confused already, just make sure you come back to this key so you know which letters are going to represent which genotypes and allelic frequencies. The best way to do Hardy-Weinberg is to actually practice it and see it in a practice problem, so let's go ahead and get started. This practice problem comes first. In a population of humans, 16%, or a frequency of 0.16, are not able to roll their tongue. We're going to deal with frequencies to do these calculations, so you want to convert percentages to frequencies, or that decimal version of the percentage. In the population, 16% are not able to roll their tongue, which means they are homozygous recessive for tongue rolling. Using the Hardy-Weinberg equations, determine the frequency of homozygous dominant tongue rollers in the population. So in order to figure out how to solve this problem, we need to know what it's asking. The frequency of homozygous dominant genotypes in the population, if we look back at our key, is P squared. So we're solving for P squared. We already know something in this problem though. So the one variable we already know is Q squared in this situation. That is going to be a frequency of 0.16. The next variable we know we can figure out by just square rooting Q squared to get Q, and that'll give us 0.4. Now we can use that first equation, P plus Q equals 1, to determine P. See, now we're just doing basic algebra to get the variables that we need. So in order to determine P, we rearranged some of the variables to solve for P. And 0.6 squared is 0.36, and that is our value for P squared, or the frequency of homozygous dominant tongue rollers in the population. Let's look at one more easy practice for today. Let's say we have a population of rabbits. Some are brown and some are white. The brown allele is dominant, the white allele is recessive. So brown rabbits can have big B, or big B, little b. White rabbits can only be little b. Know that the frequency of the big B genotype is 0.35. So let's try to figure out the frequency of heterozygous rabbits, the frequency of the big B allele, and the frequency of the little b allele with this information. You know that the frequency of the big B genotype is P squared. If P squared is 0.35, we can take the square root of that and get 0.59. From there, if we know P, we can easily solve for Q. After we solve for Q, and we're looking for the frequency of the heterozygous rabbits, all we need to do is get 2PQ. So 2 times P times Q. That's 2 times 0.59 times 0.41 would be about 0.48. The frequency of the big B allele, we already found, that's 0.59. The frequency of the little b allele is 0.41. A fish scale color has been connected to a gene with 2 alleles. Individuals with 2 recessive alleles show a blue phenotype, meaning they have blue scales, whereas those with 2 dominant alleles, or heterozygous genotype, show a black phenotype. In a population in the Caribbean, 1 in 2,000 fish show blue scales, so we already know it's not very common. Assuming the population is in Hardy-Weinberg equilibrium, what is the frequency of carriers in the population? Carriers, again, are going to be heterozygous individuals. So what are we looking for? Let's put it in terms of Hardy-Weinberg. 2pq is going to be big A, little a. So we're looking for the value of 2pq, or the frequency of heterozygous individuals in the population. What do we know already? Well, we know Q squared, which is the frequency of the homozygous recessive genotype in the population, because we know that fish have to have 2 recessive alleles in order to be blue, and 1 in 2,000 fish are blue. So that's little a, so that's Q squared. So 1 divided by 2,000 is going to get us our frequency, which is really small, 0.0005, teeny tiny frequency. So from there, we can start just to calculate the rest using basic algebra. Well, let's get Q first. Get the square root of 0.0005. That's going to give us 0.0223. Again, I've rounded a little bit here. And then we can rearrange P plus Q equals 1, which is one of our formulas. It's on your formula sheet. 1 minus 0.0223 equals 0.9777. And that is our P value. So now all we need to do is take 2 times P times Q to get the frequency of carriers. And we multiply all those values together and we get 0.0436, which is the frequency of heterozygous individuals in our population.
So phylogeny is the study of evolutionary history and how organisms are related to each other. And a lot of what we talk about in evolutionary history and phylogeny is the idea that all organisms come from a single common ancestor. Now, when we talk about using phylogenetic trees, which are models to show these evolutionary relationships, sometimes we're actually going to be using cladograms. You might have been taught that cladograms and phylogenetic trees are the same thing. There's actually a few differences between them, but knowing all the precise differences between the two is not something you're going to encounter in high school biology or even AP Bio. So a cladogram might look like something you see here that's depicting the evolution of different types of plants, or it could be very complicated like this phylogenetic tree we see on the other side, which is all the members of the Mycobacterium genus and how they're related to each other based on biochemical evidence. Usually in a phylogenetic tree, this is the branched one you see right here, the distance between 2 individuals is going to relate to time and how closely related certain species are. Cladograms, on the other hand, are mostly based on shared traits, not DNA or other biochemical evidence, and they're not going to be drawn to scale. And not all phylogenetic trees are drawn to scale either. But for the purposes of this video, we're just going to be talking about evolutionary trees in general, and you'll be able to see them, how to interpret them, and how to create your own. So like I said, these are tools or models used by scientists so we can compare evolutionary history or relatedness between different organisms. So if it's a simple cladogram like this, we're going to be grouping organisms based on the things they have in common. And usually when we're creating trees, if we're going to be doing this on our own, we want to create one that is the simplest explanation for how these traits could have evolved. That's called maximum parsimony, and it means that in most cases, a trait will usually evolve just one time instead of multiple times on multiple different branches. So if we go by that, then we want to create the simplest possible explanation for how these organisms could be related to each other. And don't worry, you'll see what I mean in just a little bit. So in the past, phylogenetic trees used to be created just on shared traits or morphological features, physical features that organisms had in common. Now we use lots of biochemical DNA evidence, similarities of different proteins, so the amino acid order in proteins of different organisms. And that's the data that's going to go specifically into a phylogenetic tree. And we continue to refine these trees as we gather more evidence. And when we say they show relatedness, we're showing how recently 2 different species groups had a common ancestor. So if we look at this tree right here, again, ignoring the branch lengths for now, we see that each end point on the tree, A, B, and C, that's going to represent a different species group. And the last time on this tree that A and C shared a common ancestor was at this branch point here on the tree. Sometimes on trees, you'll also see these synapomorphies or shared derived traits. And these can be marked by little hash marks on the tree. And any organism that appears after that trait is marked on the tree has the trait or continues in their evolutionary history with that trait. So for example, on these ones, long wings is a trait that both species B and A have, but species C does not have. And a lot of these times, these trees will be rooted, and what that means is there's going to be this sort of continuing branch or part of the tree, and it's actually the root of the tree, and that is in reference to LUCA, or the last universal common ancestor, which we think all living things share. So if we're looking at a tree like this that has this type of branching pattern, remember that continued root, that is in reference to the last universal common ancestor. And then this circle mark here, that is a speciation event, or an event where these two different groups branched from each other. And as time goes on a phylogenetic tree, it will continue from where the ancestor arose. A lot of times these are not drawn to scale along a period of years or millions of years. Okay, so let's look at this plants one more time. You can see now we have an example where these traits appear on the cladogram. And so we see that mosses, ferns, pine trees, and flowering plants are all on this tree, and they all arose from one common ancestor. Now, the last time mosses and flowering plants shared a common ancestor would have been at this branching point down here. And then vascular tissue, this is a shared derived trait that only ferns, pine trees, and flowering plants share. The evolution of seeds occurred here at this point in the tree, so only pine trees and flowering plants have seeds, and then of course only flowering plants have flowers. Everything that branches from before this mark on the tree does not have that trait. And classification systems have changed so much over time. Scientists used to just group organisms by what they look like and physical similarities that they had in common. Now we have much more reliable DNA evidence, other biochemical evidence like the order of amino acids for different proteins and organisms that we use to build evolutionary trees. Scientists classify organisms into different groups or categories, and this includes all organisms that are currently living today that we know about and every organism that we've discovered from the past. And they base these categorizations and hierarchies on similarities in their morphology or physical structures, on their developmental stages, so embryology, and they use information like biochemical data, but we can draw conclusions about organisms that lived in the past based on data we collect from the fossil record. Now, there's been many different classification systems over time. It used to be just two groups, plants and animals, and then it got a little bit more specific. In the past, there's been the kingdom structure. Now we organize all living things into three major domains: bacteria, archaea, and eukaryotic, or organisms with a nucleus. So let's talk about these domains a little bit more. Bacteria are super prevalent all across the globe, and most bacterial species don't cause disease. And there's a lot of them that are really beneficial, that help us create food like cheeses and yogurt, and others that help us produce medicines. Archaea is its own domain, and these are also prokaryotic organisms like bacteria. They do not have a nucleus. Many archaea are known for being extremophiles, meaning they live in really extreme environments, so really hot environments that are extremely acidic, or really harsh environments. And then eukarya includes anything with a nucleus, so that's plants, animals, fungi, protists, which we'll talk about in a little bit. So these levels of classification are the different categories that we organize organisms into, and different species may have slightly different levels of classification depending on what you're looking at. Some plants don't go exactly with all of these levels and sublevels, but domain is the largest, and then it goes kingdom, phylum, class, order, family, genus, and species. This is the typical categorization for all living things. And genus and species those are the two most specific categories for the most part. Sometimes we can have a subspecies. And we use these two categories to create the scientific name for an organism. Now, a scientific name follows the binomial nomenclature format, and every organism ever discovered is given a scientific name that is two words. The first word comes from the genus group, and the second word comes from its very specific species group. So its full species name or full scientific name is those two parts put together, the genus and the species. And so it's always two words, unless we have a subspecies. And it's always kind of Latin sounding. It's always in italics. The first word is capital, and the second word begins with a lowercase letter. So some common organisms you might know, here are their scientific names: Gallus gallus, that's a chicken; Sus scrofa, that's our boar here; Entamoeba histolytica, that is an amoeba; and Toxicodendron radicans, that is poison ivy. And anytime a new organism is discovered, they are given a new scientific name. What's cool about these is that they are the same all across the globe. Everyone can identify, for example, a chicken as Gallus gallus. So we don't have any language barriers there. Now, within eukaryotes, or organisms with a nucleus, animals is probably one you're very familiar with, plants, fungi, and of course protists. They used to be a kingdom themselves, but they're actually so different that we have a bunch of different categories for them. And so going backwards through these categories, protists are usually unicellular. There's some multicellular protists like kelp, but they are eukaryotic, so they do have a nucleus and other complex organelles. They have a bunch of different adaptations for movement, like cilia or pseudopods, as part of their cellular structures. Fungi can be unicellular, like yeast, or multicellular, like these mushrooms here. And they are organisms with cell walls, so each of their cells has a cell wall like plants do, but unlike plants, fungi have chitin, which is a special material that helps it maintain its structure. All fungi are heterotrophs, meaning they have to get their food from another source. They can't produce it themselves. And so fungi typically are decomposers. They can also be parasites as well, and so they'll break down dead organisms for their nutrition. Plants are autotrophic, so these are typically green. They will be performing photosynthesis in order to get the food that they need, so they use sunlight energy and convert that into glucose or other organic compounds. And within the plant kingdom, we have things like mosses and ferns, gymnosperms or conifers, and angiosperms or flowering plants. And then, of course, animals here, these are multicellular organisms. They do not have a cell wall, they are heterotrophic, and they consume their food in a variety of different ways. It can be herbivores or carnivores or omnivores. So over time, there have been many theories about how life first appeared on this planet, but some of the most important experiments that have been done to figure out what was going on include Louis Pasteur's experiments that disproved spontaneous generation, or the idea that life could just appear out of nowhere. And his actual version of this experiment happened with chicken broth and an S-shaped flask to show that bacteria couldn't just appear out of nowhere either. In 1955, one of the most important experiments in our understanding of how life could have arisen on this planet was done by Miller and Urey, and they predicted that if energy was introduced to certain elements and gases, such as a burst of lightning, so they replicated that with electricity, that organic compounds could appear. And they were successful in generating the precursors to some of our organic compounds that are essential to life itself. Now, they didn't create life in this experiment, but they formed precursors to these molecules that are the basic ingredients for life. So, through this experiment, we were able to support the idea that organic compounds could have formed in the conditions that were present on early Earth. So Luca, like we mentioned in the last video, the last universal common ancestor is this hypothetical organism that's believed to be the common ancestor of all life on Earth that arose billions of years ago. There's lots of evidence that unifies all living things. For example, all living things have the universal genetic code containing DNA or RNA. We see common metabolic pathways like glycolysis in virtually all living things, as well as some physical similarities in the shapes of cells across all domains. Remember that we have prokaryotic cells without organelles or membrane-bound organelles and eukaryotic cells with them. And scientists believe that eukaryotic cells arose through a process called endosymbiosis. So this theory of endosymbiosis describes how larger prokaryotes could have engulfed smaller ones, and those smaller prokaryotes formed a symbiotic relationship with the larger ones and later became things that we know as the chloroplasts or the mitochondria. And this is supported by the fact that the mitochondria has its own DNA and its own membrane and could have once been a prokaryote itself. Okay, let's go through a brief timeline of some major events in Earth's history. I'm not going to hit on the 5 major mass extinctions or some of the major geologic areas in this video, but that is definitely something worth reviewing if it's covered in your biology class. I just want to mention also too that the timeline of life on Earth is huge, and this is not representative of all the major events that occurred in the evolution of different species on this planet. These are just some interesting things that happened at different points along Earth's timeline. So about 4.6 billion years ago, Earth was formed, bombarded by meteorites and comets. And then around 3.8 billion years ago, we think the first replicating molecules, which were the precursors to RNA or DNA, first formed. Then we think around 3.5 billion years ago, our first unicellular life evolves, and we have, for the first time, photosynthetic bacteria releasing oxygen into the atmosphere. But it's not until around 555 million years ago that we have many multicellular organisms in marine environments and diverse multicellular life in the oceans. Around 450 million years ago, we see arthropods moving onto lands that would later become things like our land insects, mites, millipedes, spiders, scorpions. 420 million years ago, land plants first evolved. So it's interesting to see that that is later than our first land arthropods. And then 225 million years ago, we have dinosaurs and mammals on Earth, and 65 million years ago is when the KT extinction occurs, wiping out the dinosaurs on Earth. So they go extinct, and birds and mammals are able to survive. And then 130,000 years ago, we see the evidence of the first anatomically modern humans on planet Earth. Now, we know that all organisms need energy to survive, to maintain their structures, to grow, to reproduce. But different organisms have different ways of obtaining or harnessing that energy. So a lot of what we talk about when we talk about energy in ecosystems is how the energy can flow through ecosystems in different ways. Now, we'll also be talking about how matter can flow through ecosystems, and that's what food chains and food webs can show us, is the flow of matter and energy between different organisms in an ecosystem. And we're going to begin to see how all organisms are interconnected and how, for example, a change in the number of producers in a particular ecosystem can affect the consumers at the secondary, tertiary, even quaternary consumer level. Let's take a closer look. Remember, life is organized. It's one of our characteristics of life. We know that a group of organisms of one species in one location is a population, and populations can be categorized by functions that they serve. Right here, we're looking at a food web. Organisms are characterized in different trophic levels or energy levels depending on where they feed and what they consume. Producers can generate their own food, usually from sunlight energy, sometimes chemical energy as well. Consumers consume the producers, and decomposers break down organic material from other organisms. A food chain shows one pathway of energy flow. A food web, like you see here, can show the interconnectedness of many food chains within a particular ecosystem. Keep in mind that on a food web, the arrows always point towards the consumer or the organism that is doing the eating. So here we have the rabbit arrow pointing towards the cougar or mountain lion, which means the rabbit is getting eaten by the cougar or mountain lion. He is doing the eating, so the arrow points into his belly. Important word here is an autotroph, also known as a producer, but these are organisms that can make their own food or are able to form their own organic molecules for food. Plants are autotrophic. Heterotrophs are organisms that must consume other organic compounds for food, and that means consuming other organisms. Heterotrophs are consumers. For example, humans are able to obtain nutrients by consuming other organisms or the products of other organisms. This is a typical trophic or energy pyramid that you might see. We have our producers or our autotrophs at the bottom. These are organisms that are going to get energy from the sun through chemical processes, photosynthesis, and then on the next level up, those are the primary consumers, the organisms that consume them. The next level up, we have the secondary consumers, tertiary consumers, and quaternary consumers. There's different types of pyramids too, like pyramids of biomass and pyramids of numbers. But here, what we're looking at are these different trophic levels, or levels that organisms feed at. And we want to keep in mind that at every level, we have a significant amount of energy lost. So it is inefficient. We have about a 90% energy lost every single time we go up a trophic level. So it's more efficient energy-wise to consume at lower trophic levels, but only about 10% of that energy is retained every time you go up a trophic level. I want to go back to talk about how all energy on Earth is really supported by the sun. And of course, all matter here on Earth is here to stay, and the atoms and molecules that we interact with and make up our bodies are the same atoms and molecules that were here hundreds of thousands of years ago. Think about how carbon, hydrogen, nitrogen, and oxygen all combine and recombine together and are passed through different food and matter systems. But analyzing the flow of energy and the cycling of matter within an ecosystem is an important part of biology. So this diagram is something that you might want to consider. These plants are going to use carbon from the atmosphere to undergo photosynthesis and create glucose and oxygen, which are our ingredients for respiration. But if we think about if more snails are added to this container, what effect is it going to have on the plants in the container? Eventually, if we add more snails, we would see more carbon dioxide, and more carbon dioxide would lead to more plant growth because the plants could take in more carbon dioxide to perform more photosynthesis. Now, there are lots of ways that matter and energy are cycled throughout the planet. The carbon cycle is one example of that, and there's lots of different processes where carbon, that element, is converted or passed between different organisms or between organisms and the environment. So from the air, plants and other things like phytoplankton in the water can take in carbon as carbon dioxide and use that in the process of converting sunlight energy into organic compounds. So the carbon dioxide in the air goes directly into those organic compounds or things like glucose. And then all living things perform respiration, but in the process of respiration, we use that glucose to generate ATP, which is another molecule organisms use for energy. So that process is respiration. So if we follow the path of carbon here, we say that carbon can come from the atmosphere into photosynthetic organisms like trees and then can be consumed by other organisms. So when So when animals eat plants, they're taking in carbon that way, and then they're exhaling carbon dioxide after the process of respiration. Now, not all the carbon leaves their bodies. Eventually they will die, and after death, the carbon can be returned to the environment through the help of decomposers who will break down the carbon in dead organisms' bodies, and that decomposition process will release carbon either back into the soil or back into the atmosphere. Another way carbon can enter the atmosphere is through combustion. So the combustion of fossil fuels also releases lots of carbon into the atmosphere. And when there's lots of carbon in the atmosphere, it actually can be taken up by the waters in the ocean. And sometimes we have lots of carbon going into oceans, and that carbon actually turns into carbonic acid in solution in water, which leads to lots of ocean acidification. So the more carbon in the atmosphere, the more the oceans can take in, and the more acidic the oceans get, which is not always great for the ecosystems there. Now, if we go back to our levels of organization, just as a reminder, remember that a population is a group of organisms of the same species living in the same area. Typically, community is one level up or one level of organization greater than population, and it involves all the species that interact and live together in a particular ecosystem or habitat. When we get to ecosystem, that's when we're talking about the biotic and abiotic factors together, but think of population as one species, community, several different species interacting together, and then ecosystem, all of those species plus the non-living factors as well. So today we're going to be talking about factors that influence population growth, different ecological relationships. You might have heard of types of symbiosis like mutualism, parasitism, commensalism, and of course the role of different non-living or abiotic factors. In one ecosystem, we have lots of biotic, living, or abiotic factors. And an ecosystem is shaped by the interactions between these non-living and living factors. In every single environment, there is competition between different organisms and interactions between different populations. And often within one ecosystem or environment, there are natural checks in place that keep populations and ecosystems relatively stable over hundreds or thousands of years. Though sometimes we might see certain individuals that have what looks like unlimited or exponential growth, most of the time, most populations will reach what we call as a carrying capacity within a particular environment. Because there are limited resources within a particular environment, like food availability, water, space, at some point those organisms are going to reach the maximum population that that ecosystem or environment is able to sustain. And even though that may fluctuate over time, a population may be relatively stable once it hits its carrying capacity. If we see a sharp decline like this, it's probably because there was an introduction of a disease, or a new predator, or an invasive species. All of these biological molecules can be limiting as well. So the number of organisms any particular habitat or ecosystem can support is dependent on the availability of certain resources within that environment. For example, energy, water, oxygen, nutrients, all of those are going to have an effect on how many organisms an environment can support. In general, there's lots of factors that are going to influence the growth of the population, but we'll see growth typically when the birth rate is larger than the death rate of a population. If we don't have any limiting factors, a population can grow exponentially and it'll grow in that kind of J curve. But if we have some sort of limiting factor or a particular resource that's going to limit the population growth, we'll see a logistic growth curve. So it'll increase until it reaches that carrying capacity and kind of level out. There's also a lot of other factors like community structure, interactions, energy availability that are going to determine how those populations will continue to grow and survive. This question just gives us a random population and shows us how the population is growing over time. In this graph, you'd have to estimate where the population's growth rate is the highest. Well, it's going to be at a certain point in time. Can you see where the slope is the greatest for this particular population? That's where the growth rate will also be the highest. Okay, when we talk about the interactions of communities, though, there's different types of interactions like predator-prey or predation interactions, but we also have competition, different types of symbiosis.
Organisms can interact with each other in various ways. Mutualism, for example, is a type of behavior where both organisms are going to benefit. So you might think of things like a bee and a flower. Both are getting advantages here. The bee is being able to get the pollen in order to create the honey it needs. The flower is helped with its reproduction by the distribution of pollen. Another type of symbiotic relationship is parasitism. This is where one organism benefits and the other is hurt or harmed. Mosquitoes, and of course the protus malaria is a parasite as well. Commensalism is a type of relationship where we have one organism benefiting and the other is neither hurt nor harmed, and these are things like barnacles that attach to a whale. Now, there's also predator-prey relationships, which can be very dynamic, and predator-prey populations can fluctuate depending on what's happening within the ecosystem. In terms of communication and behavior, a few terms you might want to know. Imprinting is when a young animal learns something specific in a particular period of time. Territoriality is where there is a protection of a habitat or territory with tactics such as aggressive behavior, songs, pheromones. Conditioning is a process that leads to learning, and we have both classical and operant conditioning. And habituation is going to be when an organism is going to show a decreased response to a stimulus over time. And ecosystems can be relatively stable over hundreds or thousands of years, even though populations within the ecosystem may fluctuate depending on certain factors.
So biodiversity in general is just the variety of life on Earth. There are countless species literally on planet Earth, and so many we have not discovered. But a lot of times when we talk about biodiversity, we're talking about species biodiversity, which is a measure of the variety of different species in an ecosystem. So an ecosystem with very few different number of species has low biodiversity, and an ecosystem with very many different species has high biodiversity. We can also talk about things like genetic biodiversity, which is the genetic differences among individuals within the same population. But for the purposes of this video, we're going to be mostly talking about species biodiversity when I say biodiversity. The higher an ecosystem's biodiversity, the more resilient it is to changes in the environment. And so we often say the more biodiversity an ecosystem has, the healthier it is. Ecosystems with lower biodiversity and just fewer parts to them are often more susceptible to things like disease. When one disease wipes out a particular population, if there's only a few species in that ecosystem, it could have devastating effects on the entire area. These areas are often less resilient to other changes as well. So why do we care about biodiversity besides saying that an ecosystem with higher biodiversity is healthier? Well, mostly it's due to the fact that humans rely on other organisms for almost everything, for products, for medication, for the supplies we need to build and construct things, timber, fibers, adhesives, rubbers. All of these products come from things that were once living. Many prescription drugs that have been developed come from wild species, and there's so many more species that haven't been studied that have huge medical potential. Other species could be indicators of changes within an environment, of toxins, for example. And when we notice if population levels of certain species rise or fall, this could be an indicator of bigger problems within an ecosystem. Now we know that every species occupies a particular niche within an ecosystem or specific role that it plays, but some organisms may have a greater impact on a particular ecosystem and its stability than others. And so we call these often keystone species. The wolves from Yellowstone Park are often used as an example of a keystone species, even though there's been some debate recently about other organisms that may be just as important to the balance of that particular ecosystem. In general, we know that climates with warmer and wetter conditions have higher biodiversity. So abiotic factors can influence the biodiversity of an area as well. So if you think of a typical rainforest with very high temperatures on average throughout the year and high levels of rainfall, these typically have the highest levels of biodiversity on the planet. Then you compare that to something like the Arctic tundra. While not completely barren, there's plenty of species that are able to survive in the tundra. It's fewer than the rainforest, which has conditions that support a greater variety of life. Now, there are lots of different ways to measure biodiversity. There are different biodiversity indices, or a biodiversity index is one way to quantify the levels of biodiversity in an area. And of course, we can't go and count every single species in every area. So often what scientists do is ecological sampling, where they use a portion or a piece that's a representation of the whole ecosystem. So one of the main themes is how organisms respond to different things in the environment, whether that's a stimulus or whether that's a change in the environment like day to night or seasons. You might have covered photoperiodism, which is how plants respond to the changing length of day and the changes that they undergo, such as how leaves change in response to seasonal changes of day length. Phototropism is how plants or other organisms can grow or orient themselves towards light, photo meaning light. But there's lots of other examples of how organisms respond to different changes in the environment. You are not required to memorize every different type of organismal response, but there are a few key examples that we're going to go over. So I'm going to highlight taxis and kinesis because these are two pretty common things that we can see in experiments. These are types of movement behaviors that we're going to see in organisms. So taxis is a movement toward or away from a particular stimulus. A lot of times it's light or a food source, and it is directional. So in any type of taxis, whether it's chemotaxis chemiotaxis or phototaxis or thermotaxis, the organism is either moving toward that stimulus or away from it. A lot of prokaryotic organisms will have chemotaxis, so these bacteria that can move towards this particular chemical stimulus. But you might have seen within an experiment with fruit flies, for example, there might have been some taxis behavior if you soaked a cotton ball in a particular sugar. Now, kinesis, on the other hand, is still movement in response to a stimulus, but it's random movement, it's non-directional. So if there's high humidity within an environment with like a lot of pill bugs, they may not move around a lot because they're not maybe searching for water. But when an environment is more dry, you might see more random movement with these pill bugs. Okay, another theme here is that organisms change behavior within themselves or with other organisms in response to different cues. So, for example, meerkats are a really popular example here. They have predator warnings where they produce sounds and call out to other meerkats if there is a predator nearby. We also use this example of the fight or flight response in different organisms where, like in humans, for example, we have the amygdala that rapidly sends signals to the rest of the body. It's going to increase production of particular hormones like adrenaline in the body, prepare the body to either run and flee a dangerous situation or to fight off something like a predator or a competitor. We also see different behavioral responses in plants, whether that's producing different chemicals or over a long period of time. We see that plants that have high levels of caffeine in them, that's actually something that's toxic to insects that would be consuming them. So that's a plant's response to a particular herbivore. Now, just as a reminder, these are just examples. You do not need to go and memorize everything that I say in this video. In fact, it might be good to look at some different examples just to get more familiar with them for the AP Bio exam. But in general, we're going to see that certain behaviors will affect the fitness of particular organisms. So the more successful a behavior is, the more likely that organism will survive, reproduce, and pass that on to their offspring, especially if it is an innate behavior, one that they are born with, that they've inherited. Now, there are learned behaviors which parents can teach their offspring or offspring learn from other organisms, but often these types of behaviors are cooperative and they're going to benefit other organisms within the same species. It's pack behavior, herding, flocking, schools of fish. Okay, getting into another topic, energy flow. This is big in the ecology unit. We know that organisms have different strategies for regulating body temperature, so a type of homeostasis. Two main ones are endotherms and ectotherms, where endotherms use their metabolism, so thermal energy from their metabolism, to generate the warmth that helps them maintain homeostasis to the temperature that they need to be. Ectotherms, though, are dependent on the environment, so they can regulate their temperatures through behaviors like moving into the sun or moving into the shade, right? Like lizards here sitting on a rock, which can help them regulate their internal body temperatures. Now, when we talk about energy availability, typically smaller organisms have higher metabolic rates, they have higher energy needs, so something like a fly or a small rodent is going to have a higher metabolic rate than a larger organism like a cow, for example. And in general, obviously, organisms consume other organisms to gain energy, and a net gain of energy is going to result in growth for the organism and storage of energy and overall survival. A net loss of energy, we get a loss of mass of the organism, and then later death for the organism. Scientists like to measure communities in a variety of ways, and we can use mathematical models to investigate population growth patterns. With an exponential growth model, we assume that there's unlimited resources for the population. This is usually contrasted with the logistic growth model, or the model where we use carrying capacity. It's unlikely you'll see a problem on the AP Bio exam that asks you to just calculate population growth alone or exponential growth alone. More than likely, you'll have to apply ideas behind these two equations, or be able to recognize exponential growth as a type of growth due to a lack of limiting factors, or when our reproductive or growth rate far exceeds the death rate of a population. Sometimes we'll see bacterial populations that can grow like this, but most populations in nature will reach carrying capacity eventually. We reach carrying capacity when the reproductive rate equals the death rate. Usually, this will stay the same as long as environmental conditions stay the same. We also have Simpson's diversity index to measure the diversity of a particular community based on different species numbers and population sizes. Let's look at the predator-prey model. You should be able to recognize this as a very typical predator-prey relationship graph. But if the predator is hunted to extinction, after a short period of time, we should see a spike in the population of prey. If we look towards longer periods of time, the population of prey may actually get too large, and there may not be enough resources for the entire population. So we know that Earth's organisms share limited resources. We rely on processes like the water cycle, the nitrogen cycle, the carbon cycle to cycle out essential nutrients and molecules in and around the Earth that are essential to survival. And of course, the Earth changes over time, but humans are a huge source of environmental change. And if we look at this graph here on screen, I'll blow it up for you so it's a little bit bigger. This is the world population growth in billions is a little outdated because we know now that the world's population is estimated to be above 8 billion. But the depletion of Earth's resources and different impacts from humans and their behavior is often a direct result of human population growth. And often humans are the cause of major disruptions to different natural ecosystems. And because we rely on the natural world and organisms and things found in nature for products and for medicine and for our own survival, making sure that we protect Earth's resources and its organisms is important to our own survival. Now let's talk about these resources that are found on Earth. So renewable resources are any natural resource that can be replaced by natural processes relatively quickly. When I say relatively, generally that means within a human lifespan or at a rate faster than the rate that they were used. Non-renewable, on the other hand, cannot be replaced within human lifetime. Now, non-renewable resources come from natural processes, like fossil fuels, for example, but it takes millions and millions of years for these resources to form. So when we're speaking in realistic terms of humans and what we have access to, once non-renewable resources are used up, they are gone. There's not going to be any more for a long time, and they may not even replenish. They are a limited resource, and they may be used up by humans. So examples of these are coal, oil, natural gas. Renewable, on the other hand, we could talk about things like wood, organisms that we use as resources like fish. But it's important to note that renewable resources can become non-renewable if humans use them up too quickly. Now I want to talk a little bit about the ozone layer. Now this is an interesting case of human impact, and the hole in the ozone layer we know is primarily caused by CFCs or chlorofluorocarbons, which are chemicals that come from things like old aerosol sprays, and these have actually made an increasing hole in the ozone layer. Now the good news is that after mitigation, after some laws that went into effect banning CFCs in certain products, we have started to see a decrease in the hole in the ozone layer, which is really good. But this is the progression of the hole from 1979 to 2008. So you can see in these images, they're getting bigger. But in the recent years, we have seen data to show that this hole is shrinking. So that's some good news. But the ozone layer protects Earth from harmful radiation, UV radiation, and we know that compounds in certain products have damaged this ozone layer. One thing students often confuse is that the hole in the ozone layer is a major contributor to global warming. And those are actually two separate issues, so let's talk about both of them. Global warming is an increase in the average temperature of the biosphere on Earth. It's mainly a result of burning fossil fuels and introducing greenhouse gases into the atmosphere, which trap heat at a higher rate than normal. And alongside global warming, we see other issues like rising sea levels, coastal flooding, melting of polar ice caps, melting of glaciers, all of which can have devastating effects on the environments and the people who live in those areas. Now, there are other activities that humans can do that cause disruptions, not on such a massive global scale, but on a scale that can be a huge problem for local ecosystems. So for an example, humans often have either intentionally for a good purpose or unintentionally introduced an invasive species to an area that does harm to the native species in an environment. Now, an invasive species is a non-native species, so something that wasn't originally from that place, that comes and does harm to the local ecosystem. A really common example of this is kudzu in the southeast. It was introduced as a decorative vine, something used in erosion control, and it has completely overtaken local ecosystems, totally covering native plants, blocking out the sunlight, outcompeting native species and reproducing at a faster rate, which then decreases the overall biodiversity of the ecosystem, which we talked about in yesterday's video. Human activities can also introduce new diseases to an area. Elm disease was a tree disease that was introduced, totally devastating, and often we see more of this with the increased movement of humans and human activity on the planet. And of course, habitat change is a huge driver of species either becoming threatened, and habitat change, of course, is a huge threat to different species. Actually, habitat loss is the number one reason species are at risk of becoming extinct. Habitat loss can occur for a number of different reasons from human activity. So pollution in an area could destroy habitat. For example, certain chemicals or gases could kill off native species that different organisms rely on to survive. This can obviously come from the burning of fossil fuels, but also chemicals, runoff. Even certain fertilizers could end up be damaging to different species in an area if they're carried off into local water supplies. Deforestation, just removing a large amount of trees for a certain area, either for the use in lumber or for building roads or other things. And again, related to that is logging, monocropping, so when you're planting only one specific species in an area. And industrialization is in just any area, A is a good example of this, but as more nations become industrialized, we see more and more of this habitat change in local ecosystems. All right, you did it. Thanks so much for sticking with us. I hope this has been helpful. Make sure to give this video a like if it has, and subscribe so you don't miss out on any of the other great resources that are on this channel that'll help you succeed in your courses. I'll see you later.